Hi, my name is Athenia, and I'm here to talk to you about the stuff of science fiction. And no, I don't mean warp capabilities or phasers, even though that stuff is pretty cool. I mean tissue engineering. So by its formal definition, tissue engineering is the capability to create biological substitutes that restore, maintain, or improve tissue functions. And this has huge implications on things like wound healing, or on the less therapeutic side, biosensors. But today I'm here to talk about the white whale of tissue engineering, which is the creation of entire organs. So let's start with the basics. Engineered organs are going to have three main components: a scaffold, growth factors, and of course, cells. First, let's start with the scaffold. The scaffold is something that will provide structural support for the cells. It's something that they can adhere to and grow around to form different structures. Now, this can be anything from a natural protein like collagen or something artificial like a polymer. And in vivo, cells actually excrete this thing called the extracellular matrix, that is a network of proteins that provide structural support for the cells and also plays a role in cell signaling, which is crucial when it comes to the repair of damaged tissues. So next we have growth factors. Growth factors can actually be found in the ECM. In the case of engineered tissues, they'd be put in the media, and they help promote cell growth and development. So I'm going to do a quick demo to show you how the parts come together. Let's say in this case I'm trying to engineer a blood vessel. I'm going to need a scaffolding, some cells, and in this case it'd be endothelial cells and periocytes because they're the main components of blood vessels, and a growth factor to make sure everything goes okay. In this case it'd be VEGF, which promotes vasculogenesis. So you have your scaffolding, and the cells adhere to the scaffolding and start to form a structure. And the VEGF promotes the vasculogenesis to make sure the structure forms. In some cases, scaffoldings can be made to dissolve, so you just have the finished product left over. See? A fake blood vessel. So now that you know about the basics, I can tell you about some truly amazing things happening in tissue engineering today. Researchers are taking donor organs and removing their cells, and so there's just a scaffolding remaining, and then seeding them with patient cells. Now these organs are fully functional, and they remove the need for immunosuppressant drugs because it's the patient's own cells, but they are unfortunately not implantable yet. Another innovation in tissue engineering comes from something a little bit less expected. 3D printing actually has huge implications in tissue engineering. By 3D printing a scaffolding and then seeding the cells within that scaffolding, you eliminate the need for donors. Now, this is still a highly experimental process, but researchers at Princeton and Johns Hopkins University have created a prototype of the outer ear. A lot of progress has been made in this field within the last decade, and hopefully in the next decade we'll see a reduction in costs and implantability of the organs that are actually made, and also the complete elimination of donors. Thanks for listening.